Hello, and welcome to this Chemistry World webinar, Driving the Development of Bio-Based Polymers with Molecular Simulation. I'm Philip Broadwith, Chemistry World's business editor. For those of you not familiar with GoToWebinar, if this is the first time you're using it, the important thing is that GoToWebinar is an interactive platform. If you're having any kind of technical issues, you can contact the team using the questions box, which is at the bottom of your GoToWebinar panel. By default, that's usually on the right-hand side of your screen. If you do miss any part of the presentation, or you just want to watch it again in your own time to pick up on some details or links or resources or whatever, then we will be sending out a link to everyone in a few days' time. And to say thank you to everyone who's attended the webinar live, you'll also get a certificate of attendance in that same email. Today's webinar is about using large-scale molecular simulations to reduce the development time and cost for bio-based polymers. Moving away from fossil feedstocks to more renewable alternatives is a key part of the chemical industry's transition in the coming years, but that shift in materials presents some serious challenges to developing alternative materials that fulfill the performance requirements expected of their petroleum-based counterparts. Molecular simulations are a great way to assess and model the behavior of bio-based polymers, allowing the teams to exercise their existing expertise in formulating new materials and to do that quickly and cost effectively. We have worked with Schrödinger to bring in this webinar, and we think it's packed with information that you will be interested in from real experts in the field. Today's speaker is Andrea Browning. She is the product manager for polymers and soft matter at Schrödinger. Before joining the company, she worked in the aerospace industry at Boeing and has a great deal of experience in simulation and in linking that simulation to industrial decision making. I can see, Andrea, that you've got your camera on. I'll just check that we can hear you as well.
Yes, hello. Thank
Excellent.
you for that introduction.
Hi. No problem. Once we've got your slides up, I'll hand over and you can crack on with the presentation. Take it away, please.
Great, thank you so much for that introduction, and it's my pleasure today to speak with you about bio-based materials, a really exciting part of our advancing chemistry future and the way that we're moving towards a new life cycle for polymers. And when we look at the life cycle for polymers, we're really focused on what can we do to bring forward new materials that are going to have a life cycle that's not as damaging to our environment and the economics that we now look at when we look at material use. So polymers and plastics are ubiquitous in our everyday lives, and they are amazing molecules and materials that have provided us an opportunity to advance in lots of areas that we hadn't been able to before their invention. But as everybody knows, we've found that that comes with a cost, and we are have seen over the last decades what that cost is, and now looking at what can we do to keep of materials while still looking at really what's that overall life cycle cost. And so as we look at bio-based materials as one tool in that tool set of, okay, how do we address polymers and plastics and their overall life cycle, that is, bio-based materials cover a wide area of what can be used in the future and what's being used today in a lot of different areas. And these bio-based materials, when I use that term, using in kind of the broadest sense of any kind of polymeric plastic material that has some root in a living organism. So that could be from really traditional kind of saccharide-based polymeric materials, like where you're using the saccharides that are coming from polysaccharides that are coming from natural materials, and then using that to make something new, or using the fundamental building blocks from some of these natural organisms to make new monomers that then you can then polymerize and make into new polymers. And all of those have the base in a biological entity that then we're kind of reusing in a new way. And and that's providing us both on the production side a new way of getting that material, and also on the degradation side, giving giving chemistries that inherently have some degradability to them, and perhaps some recyclability built into their use. So kind of expanding out what we're thinking about when we're thinking about the design and the use of these polymeric materials. So with that kind of broad kind of space that we're talking about here with bio-based materials, and that driver of like, we want to look at where these polymers, where these plastics, where they're coming from and where they're ending up as important features, and whether these are going to be successful materials. We see this as a global initiative, and I think everyone is familiar with the different initiatives happening where we're across the world, where we're looking at from the EU to the US and across the world, looking at ways that we can drive the adoption of these kind of materials, where we can again improve that life cycle that's happening with these polymers and plastics materials. So we understand that these are important materials that we want to have take the place of traditional polymeric materials in a lot of instances, but that drop-in replacement can be difficult. So when we look at development of new materials, if we just kind of look historically across different material spaces, that could take a long time. That can be 10 to 20 years. And I think anyone working in product development or material development sees this every day, that the materials that you're using today in new advancing products are very often things that were developed decades ago and perfected decades ago, and then the products kind of catch up to that. Now, as we're pushing forward new products, we're trying to accelerate that material cost. And when we have this kind of overall push towards new kinds of materials for other kinds of reasons, so like this push towards bio-based polymers for recyclability, sustainability, and the source issues, then we're looking at how can we shorten that time frame? How can we shorten the time to market and the time to adoption for these kind of materials? So we have to look at different ways that we can understand what the connection between that chemistry and the properties, and how those properties can be taken advantage of inside the product. So then digital chemistry comes into play as a powerful tool to allow you to make these kind of connections. And so we have these external trends that are pushing for this rapid development, and we want this rapid development in a timely manner. We're combining that with increasing computing speed. So we have the GPU resources. We now have not only that just in a single GPU, but the ability to burst onto the cloud. So we have these resources that now are accessible by more and more people and more organizations in a manner that not only takes advantage of the actual compute power, but some of the kind of business considerations that go into everyday decision making of like, can you burst onto the cloud, can you get resources within a manner and in a time frame that makes sense for different kinds of projects. So these kind of advancements that we have both on the technology side and then the infrastructure side give us new capabilities and new ways of looking at simulations. So within Schrödinger, we take advantage of that and focus on the machine learning and advanced physics-based modeling to provide the right kind of tools to study these kinds of materials and to get information and knowledge that is useful in these development kind of projects. What I'll be speaking today will be mostly about the physics-based modeling within bio-based polymers and materials kind of research. But this overall fits into where we see computational materials playing a role in the future of materials development. When we look at physics-based modeling and how that differentiates from machine learning and other kinds of activities that you might have, with the physics-based modeling, we're really grounded in the first principles of atomic and molecular interactions. So we're describing the way these basic building blocks interact together, and using that as you build out the molecules, as you build out the polymers, we can understand how those interact together, and then make predictions on how those larger scale materials are going to behave. And so with the validation of those different techniques and understanding of where their accuracy, what their accuracy is, we can then use them with confidence in making decisions. And then as we combine that with machine learning and expand out and provide more insights by scanning a larger chemistry space, we have even more powerful capabilities. And then finally, and I won't speak about it too much today, but I did want to mention it as something that becomes a core pillar to the overall digital chemistry strategy, is the informatics and the collecting of information across different teams, and then sharing that information from simulations and experimental data side by side, and connecting across those teams so that you're making the collaborative decisions within these chemistry and material space. So now let's get into bio-based polymers. And so when we talk about computational chemistry and simulations as a general technique, we're speaking about how these atoms and these molecules interact together, and then predicting some property. But that's really high level. We want to connect that down to a particular problems of interest, and by making that connection down to the problem of interest and that material of interest, that's where you really make those core decisions about what kinds of techniques you're going to use, and whether how you're going to build the models, and how you're going to explore these kinds of interactions. So when we look at bio-based materials and the use within different kinds of products, we might be talking about different kinds of problems that would be arising in different development projects. So it could be something where you have an existing product and you have an existing system, and you're continuing to develop that on. And so you're looking at, say, changes in the formulation. Maybe you need to select a different cross-linker to impact what the glass transition temperature is going to be. So that's going to be looking at your existing chemistry, looking at different kinds of cross-linkers, and making simulations that are going to give you a good prediction of the glass transition temperature. We could also think about new chemistry design. That might be something new chemistry design or new formulation design around a new chemistry. So there you might be screening larger libraries for multiple properties to understand what kinds of performance you have, but also what kind of risk you might have. What are the properties where that you just need them to stay stable? You need to make sure that they're not going to go down. So it may be about screening different kinds of libraries for understanding that interaction and that performance in a particular case. So when we look at the adoption of computational chemistry techniques, we're connecting what is going to be the particular problem in a particular area, and making sure that we're connecting that back to what's going to be valuable information in the overall development and design process. Once we make that connection, that's when we have then the particular technique that's going to be most applicable to those particular kind of questions. Maybe multiple kinds of simulations that need to take place to predict a different property. And with molecular simulation, we have kind of three core areas that we focus in on, and that's electronic kinds of simulations. DFT is our standard technique, and then we have atomistic simulations and mesoscale simulations, where we coarse grained out the atomic structure to look at those on a larger scale features. So once we have what our property of interest is, what our material that we're going to try to capture, then we can focus in on what's the right technique to answer those kinds of questions. And so today I'll be mostly speaking about the atomistic and the mesoscale level, and making connections to what kinds of simulations that you can do and kinds of information that you can get for these bio-based types of polymers. So one of the first questions that often comes up when we're talking about bio-based polymers is those really traditional kind of pulling from a natural source and just taking those and processing them in some way, but not chemically altering them too much. And that gives you these polysaccharide kind of polymers. And in those kind of polymers, we have these sugar units that are being linked together in a chain. And those have really distinct structure. They are polymeric materials. We have a monomer and monomers that are linked together in a certain way to make these longer chains, these macromolecules. So they are, by definition, polymers, but they have really distinct structures as far as the way those units connect together and the way that they form these chains, and there's a specific geometry within those sugar rings plays an important role in what it is, and so and the way that they perform. So the difference between say cellulose and amylose can be very distinct inside the way that the torsions on this particular chain arrange, and you can get different kinds of structures depending on that. So when we look at a natural source kind of polymers, one aspect to consider is how are we going to construct them, and are we getting not just chemistry as far as what atoms are connected to what, but are they in the right conformation that is representative of their state in the actual product. And so having the right kind of builders that builds up from those sugar units, from those sugar rings into the longer chains, and building different kinds of crystals, building amorphous or semi-crystalline kind of structures, looking at different kinds of fibers, but making sure we can build the right kind of state for those particular natural source polymers, then allows us to get better capturing of what those properties are going to be for those particular kinds of materials. So what can we do with some of these kind of polymers? I have an example here with amylose. So that's connecting that sugar polymerized sugar chemistry in a particular type of it, and making a polymer chain. And in this case, we did kind of a short polymer chain, but you still see the kind of helical structure that you see in this particular natural polymer. And then having those amylose chains together, so you have a bulk amylose system to understand what's going to happen with that bulk amylose structure. So connecting from the building blocks of the sugar rings, connecting those in the right kind of geometry to get the helical amylose structure into that bulk simulation. And once we have that bulk densified amylose structure, then we can look at things like glass transition temperature, and not just of the bulk amylose material, but we can also load in water. So you see the plot on the right, we're loading in water at different temperatures, and so that's providing, as you increase the temperature, this structure is going to uptake more water. And so you can see the uptake of the water is going to increase as you increase that temperature. And then once we have water loaded into our amylose structure, then we can look at the glass transition temperature of that water amylose structure. And as expected, as you add water into your system, then the glass transition temperature decreases. And you can see that, again, going back to the plot on the left, that as you add water into your system, your glass transition temperature goes down. And then we also, after you get to a certain point, you can compare to experiments and see that the glass transition temperature depression that you would expect from the simulations, you see that in the experiment. So you see that decrease in the glass transition temperature with that addition of the water. So if taking this kind of base approach, we can look at screening and optimizing bio-based polymer formulations now. And by screening many different types of formulations, you can look at understanding what's going to be the best formulation for a particular kind of use in less time and ultimately at a reduced cost. So in this particular case study, what we were looking at is mixing together a natural polymer, a polysaccharide-based polymer with other kinds of polymers, and in this case, we're going to use polylactic acid. And so this mixing of polymers together, anyone that's worked in polymer formulations will know very well that making these alloys of different kind of polymers together gives you that way of tuning your properties in many cases. Mixing together two polymers may not be an easy thing to do in many cases, but it gives you that nod to turn of like increasing or decreasing different amounts of polymer so that you can get the properties that you need for a particular kind of application. And you might be tuning on, say, plasticity versus mechanical properties, but you're tuning for the right properties by mixing these different kind of components together. So if we can screen for those mixtures as you're developing these kind of formulations, so that gives you an advancement on what you can do, and then you can use your experiments to focus in on what's going to be the best mixture for your particular kind of application. So in this particular case study, we looked at a number of different polysaccharides and a number of different kind of concentrations to understand what's the trade-off as you move between different polysaccharides and then different percentages. So as I mentioned, we used in this particular case study polylactic acid and different polysaccharides. So for the polysaccharides, we have cellulose, chitosan, and chitin. And looking at how the polylactic acid, as you take polylactic acid and you mix in different amounts of these polysaccharides, what happens to a suite of different kind of properties. So this is something, as you can see, the image from the literature on the right is something that is currently done, that you can experimentally mix these kind of materials together in small percentages and look at what's happening with this kind of behavior. So here we're pre-screening ahead of what can happen with these different kinds of mixtures of polymers together. So one of the first questions to ask is, when we densify these kind of structures, are we getting the right density? So here we're looking at a densified simulation and what is the density of these mixtures of polylactic acid with different polysaccharides. So on the very left most point of these plots is going to be the polylactic acid, and on the very rightmost at the 100% is going to be the different polysaccharides. And so we can see that the polylactic acid density and the different polysaccharide densities align with experiments. Chitosan is an interesting example. You can see the experimental density is in bold as it makes very large voids in its structure as it is being formed naturally. So in this case, our density is higher than that because we're on a kind of a nano scale, so we're getting that kind of chitosan wall type of density, but we do see that the chitosan has a more larger free volume compared to the other polysaccharide material. So we can look beyond just the bulk density. We can look at kind of structuring that's happening within these kind of materials. And so one thing that we see is that when you mix the polysaccharides and the polylactic acid together, you have structuring, and that's happening, phase separation that's happening within these kind of systems. And so the polysaccharides is kind of separating out from the polylactic acid, so that you end up with a region that's going to be dense in the polysaccharide and a region that's going to be dense in the polylactic acid. And we can see that from on the right-hand side, you'll see the density plots, and you can see where those regions are polysaccharide, where the regions are polylactic acid. And we can also see that in the energetic measurements. We can see that the energy favors not mixing of these kind of components. And so we can look what's happening within the different structure beyond just that they're separating from each other, what's happening to the polymer chain. So when we look at the strands of the polysaccharides in the polylactic acid, there's two main measures that we explored. One was the end-to-end distance, one is the persistence length, and as you increase the end-to-end distance and the persistence length, what that's telling you is that your chain is becoming more elongated. And so as we increase the amount of polylactic acid, it's actually moving right to left on these plots, we're getting more elongated chains. And so you can see that both in the persistence length and the end-to-end distance, the chains become more elongated for these polysaccharides as they get into a lower amount of polysaccharides. So as the amount of polylactic acid increases, so as they become the polylactic acid becomes the dominant material, you get these more elongated chains. The polylactic acid, on the other hand, stays fairly similar as far as its overall chain structure. So the polylactic acid matrix is kind of staying the same, but these polysaccharides are changing what their strand behavior is. We can also, one thing that we can do within the simulations very easily, that's more challenging experimentally, is look at detailed interactions. So you can think of these plots, these are radial distribution functions. They're giving you an idea of what's a local order of different chemical units. So it's a bit like having X-ray diffraction at your fingertips for all your kind of materials that you might be looking at. So here we're looking at how the oxygen in the polylactic acid, how those units are arranged next to each other locally in the different amounts of polysaccharide. And then in the middle part is the OH groups from the polysaccharides and how those are arranged locally to each other as you have increasing amounts of the polysaccharides. And what we see is that we have those increasing interactions of the different kinds of units as we change the amount of the polysaccharide within it. So it gives us a way of looking really in detail as these chains are lining up, as we're going into more and more polylactic acid and seeing that closer and closer interaction. We can also look at free volume and understand what kind of space, what kind of free space do we have in these systems. And so there's a lot of detail in here about different kinds of pore volumes, and these are kind of individual snapshots of what is happening with your pore space. But one thing I wanted to kind of highlight is if looking between the different saccharides, say at 100% polysaccharide structures, if we look at the different saccharides, we have different amounts of free volume within these systems and different amounts of pore sizes. So if we look at the pore size distribution that's shown here, we look at the chitin, and then we do see those larger pores that develop in that chitin structure. So you can think of that the chitin is actually, the reason its density is lower is because it is making these larger pores within its structure. So now we have a lot of information about the structure and what's happening within these kind of natural polymers, but how does it impact on the properties? So the first property we're going to look at is glass transition temperature. And so we could take, again, all these structures, the homopolymers and the mixtures, and look at what the glass transition temperature is by cooling the structures and looking where that transition happens. And so for the polylactic acid and the 100% polysaccharides, we get a good capture of what that glass transition temperature is. In the mixtures, we have phase-separated systems, so we would expect 2 TGs. What we see in the simulation is kind of the mixture together, so more of a composite TG because of the way the rate that we're doing these glass transition temperature simulations. But you see that simulated TG aligned between the 2 homopolymers. So it's getting that capture of that mixture percentage of those 2 components. And well, we can also look at mechanical properties. So we can look at both the transition from elastic to inelastic. So where does it deviate 2% from a linear point in the tension and in shearing, and we can look at yield stress. So where's the maximum in a stress strain plot, and we can look at what's that initial modulus in a stress strain plot in both uniaxial and pure shear kind of conditions. And so by sampling all these different kind of conditions, uniaxial and pure shear, and we can look at these different kind of measures, we can see that as expected, these polysaccharides are providing an increase in those mechanical properties. You are increasing your yield stress and your modulus with these kind of materials, increasing what kind of load that they can take, and you can see that increase between just the polysaccharide itself compared to the polylactic acid, and the mixture is kind of aligning in between that. And we can see as those develop, as you're moving between the different mixtures, you can see what's the appropriate kind of mixture for a particular kind of yield stress. We can also take these polymers and we can add water, as we did with the amylose case. In this particular condition, we're using 100% humidity and loading water into the systems, and you can see that the polysaccharides that take much more water than the polylactic acid, and then you have the composites, the mixtures lying in between as far as the water uptake. And that water, we can again look kind of structurally, where is that water going? We can look as a visual representation, where is the water going? Looking here, you can see the full system. Here we've taken out the polylactic acids. You can see better where the water is going. You can see the little white and red here are the water, and the green are the polysaccharide chains, and we can look at where the water is going. We can look at that kind of numerically. We can see, okay, well, what happened to the density as far as the cellulose density as we hydrate it? We could see, okay, there's a change in the amount of density that we have in particular locations. And we can look really structurally, again, kind of think of it as an X-ray kind of pattern. What's happening with the association between different OH groups, say the water and the polysaccharide, what's happening within these kind of different systems, and look structurally where's the connection points between these different systems, and look at, say, different kinds of hydrogen bonding that might be happening within these networks. But we can overall see that the water is going primarily to the interface between the polylactic acid and these strands, and we can see there's also clusters that form within the polylactic acid, and understand where that water is going and why it's going to different places. So we know where the water, how much water is going into the system, where is it going, and we can now look at what's the impact on different kinds of properties. So if we look at glass transition temperature, we can see that as expected, as you load water into the systems, the glass transition temperature decreases. So similarly, for the yield stress, we see that decrease in the yield stress as we load in the water. So we have a decrease in the overall composite performance with that water being loaded into it. So that was a lot of data and a lot of simulations that were happening for these mixtures of polysaccharides and polylactic acids that were being connected to different kinds of properties, primarily thermal and mechanical, as well as understanding structurally what's happening within these different kind of materials. If we look at a formulation kind of project, where if our primary of interest is what's happening as far as structurally, like how are these systems phase separating, we can focus in directly on that length scale and go directly to the mesoscale level and perform just the simulations that's going to give us the information that we need for that kind of structural phase separation. So as an example, here's polyethylene glycol and polylactic acid as an example of the way that you could perform these kind of simulations. So in this case, we're changing the amount of polyethylene glycol compared to the amount of polylactic acid, and looking at the phase separation that happens as you change those relative ratios. And so as you mix between these different systems, it's well established in the literature that you will shift between miscible and immiscible within the system as you add more PEG into the system. And so what we can see is that we can replicate that within the simulations. So the way that we did this was using automatic parameterization. So we're getting the coarse grain parameters with some automatic parameterization. So we're connecting to the atomistic structure and say, okay, we want to map the system in a certain way, and then we'll run atomistic simulations to directly extract what the appropriate parameters for our coarse grain simulation would be. And then from that, as I showed on the previous slide, you can look at, once you have those parameters, you can start to look at different compositions. And so it's kind of screening your composition space. So here we just did 3, but you can see that phase space start to become apparent between the different kinds of compositions. And in addition to just visually looking at that phase separation, as we did with the polysaccharide polylactic acid kind of case, we can look in more detail, and in this case we actually pulled directly the sac scattering, and so getting the simulation sac scattering pattern that you might expect, so you can think about that connecting back to your experiments and see what is the connection to the kind of patterns that you would expect to see in these phase separated systems. I want to move now to some examples of solution-based bio-based polymer usage. So when I think about usage of polymers, we can think about bulk polymers and the kind of polymers that we might, plastic kind of materials that we might traditionally think of. But there's polymers in lots of different kinds of formulations that provide different kinds of functionality, whether that's dispersing or whether that's providing different kinds of viscosity changes. But these polymers have a particular role in different kinds of products. And so when we talk about bio-based polymers and replacement of traditional polymers with these bio-based polymers, we have to also think about how they're going to behave in solution and how they're going to behave not just by themselves, but with these other kind of components. So one thing that we can look at is how can these bio-based polymers, how do they bind to other kinds of components, and whether you want them to favorably bind to kind of encapsulate, or whether you don't want them to bind to these kind of polymers, depends on their particular application. So in this particular set of simulations, we were looking at an amylose polysaccharide system with different molecules in water and how they were binding, and looking at as they're connecting to that molecule and as they're not connecting. So you can see in the movie on the right that the molecule is surrounded by the polysaccharides at some points in time, and other times it is not. And so we can use that kind of simulation to understand what's the binding and how favorable it is to bind for that polysaccharide to these different kinds of molecules. So as you can think of this plot as kind of a reverse, but down here with the high negative number means that it's actually favorably interacting and it's going to bind strongly, versus these lower numbers where it's not binding so strongly. And so if we're looking at a particular kind of molecule that might very favorably interact with the polysaccharide, we're going to be looking down here at that strong binding. It's going to be bound very strongly into that polysaccharide. If we want something that's very loosely bound, that may be able to release very quickly, it would be kind of over here. And so we can understand that as you're selecting kind of the small molecules that we're going to go with the polysaccharide, or the right kind of polysaccharide for your particular kind of small molecule, whether that's an additive for a particular kind of performance, or whether it might be a flavor or a scent molecule that you're exploring, then having the right kind of connections between those different kinds of molecules within your formulation will allow you to deliver that kind of molecule in the right space. And I want to move out to a new kind of types of molecules, and I've talked a lot about polymers, but I did want to expand that out a little bit, and then the overall topic of this webinar is, in fact, polymers, but I wanted to kind of briefly mention another class of bio-based materials that are also very, very relevant when you're talking about formulations, and that's biosurfactants. So you can look at surfactant material also having natural components and having those, again, primarily kind of sugar ring kind of chemistry within that surfactant space, and so giving you that ability to have different kinds of surfactants but still have that hydrophilic, hydrophobic kind of connection within your system. So as we move into kind of biosurfactants, the kind of architecture that you might see will be very kind of different, and you have a new space that you can explore within those biosurfactants, but understanding how they're going to structure and understanding the kind of micelles that you're going to get within these kind of biosurfactants is going to be an important part of understanding if these are going to be good replacement molecules for your traditional formulation, or if you need to have a completely new formulation to work around those biosurfactants. Now, this is just a kind of a quick snapshot of a larger study where we're looking at different kinds of biosurfactants and screening across those biosurfactants, not just for the biosurfactants performance itself, but as you change concentration and the pH of your formulation, what's happening within your system. So being able to do large-scale simulations, and many of them allow you to screen across your formulation space, so both in the chemistry and the conditions, to get you the right kind of information that you need to make the decisions about what's the right, say, biosurfactant, but also what's the right concentration pH, or if you're constrained in the pH, what's the right biosurfactant and the right composition to get you the right kind of behavior that you need for your particular kind of application. So when we've talked about these kind of bio-based materials, we talked a lot about here different kind of properties and structural kind of features that you can screen for. But another interesting aspect of bio-based materials is how they can open up the chemistry space. So if you think about the bio-based case where you're taking some kind of biomass, you're processing it in some way enzymatically or some other way to make new kind of monomers that then you polymerize in maybe more traditional fashion to get to a polymer product that you can then use. You have now these monomers that you may not have access to with some of our historic kind of pathways. And so then that with those new kind of monomers and the new kind of connection points that we can make between those, we now have kind of new polymers that we might be able to access. So with that opening of the chemistry space, though, there's the question of, well, how are we going to understand what the performance is of these kind of monomers, or what kind of monomers do we want to kind of focus on and try to get from our biomass? And so that's where a high throughput screening. So I kind of briefly touched upon that with the biosurfactant case. But the concept of high throughput screening, where you have a property of interest that you're targeting, and you have large scale simulation that are being sent to a large number of compute resources, so that in a quick short amount of time that you can screen for a large amount of chemistry. And here we did 300 polymers, 300 thermoplastic kind of polymers, looking at the glass transition temperature. And by using about 300 GPUs, we were able to get the glass transition temperature in about 12 days for all of these polymeric materials. So as we think of access to cloud resources and kind of these burst resources, we can look at projects like this of not being just purely academic kind of exercises, but rather of that we can take advantage of these compute resources, expand out, and do large-scale screening when we need it to get that selection of what's going to be the right kind of chemistry for a particular kind of use case. We can also think of expanding that out and with the machine learning kind of techniques where we can fit for different kind of properties. Here we did dielectric constant, and for another data set, glass transition temperature. So you can think of having different kind of properties that you can have machine learned models on. Whether that machine learned model is built upon experimental data or whether it's built upon simulation data, you now have the ability to say very quickly calculate different kinds of properties across different kind of monomers, and then design directly what kind of monomer is going to be the best combination of your different kinds of properties. So throughout this webinar, I've discussed about the ways that bio-based polymers, both in the polysaccharide case and then I'll say more connecting of different kinds of monomers, new kind of monomers together, could be assisted and accelerated by these computational techniques. But I wanted to go back to this slide to again say that it connects back to what is the property of interest, and making sure we're connecting to the questions that are being asked, and whether that's a continued development or new chemistry design, that you're saying what is the question of interest, and then connecting that to what are the right kind of simulation techniques to provide that information that you need. Because by making those connections, they're going to be able to either expand out that chemistry space, understand what's not going to work faster, so that you can get to what will work and less expensive and in a faster manner. And with this kind of application of simulation tools to these bio-based, a really important class of materials is bio-based, this replacement kind of activity, aligns really well with our overall Schrödinger mission, and that is to improve human health and quality of life with these new materials, and by advancing these computational techniques to enable that. And so that I hope that you saw today just a few examples of the ways that that these techniques can be applied into these really relevant problems. And throughout the problems and examples that I showed, underlying all of that, I'm showing just a few plots and a few pictures, but underlying all that is the building of the models, running of the simulations, analyzing the results, and that's where our platform comes together, and making those connections between the chemistry and the properties of interest, and making sure that you're doing the right kind of simulations to get the right kind of properties that you need. And can ultimately be connected up into our informatics platform to provide that kind of scaling across different geographies, or scaling across different projects and units that might be studying different questions. One thing that I love about polymers and working in the polymer space is the breadth of industries that we work in. And I didn't talk about any one particular one, in that these bio-based polymers are important across a wide variety of different industries. And so within our materials design group, we focus on multiple industries and look at how these kind of materials and how different kinds of materials, the simulation techniques can be applied in these different kind of industries. And so when we talk about bio-based polymers, you see a different kind of example of that, where you might have just in the traditional kind of polymer space, but also in the consumer packaged goods and in other kinds of kind of support polymer architecture, like complex formulations. But when we look at our computational techniques, and we're looking at where the different industries, the kind of questions that they're asking, and tailoring that solution for that particular industrial need. And with that, I will end my presentation and turn it back over to Philip. Thank you very much.
Hello. Well, thank you very much, Andrea, for a fascinating presentation. I think you gave a really solid overview of everything there. So just to say thanks again, Andrea, for a fantastic presentation. Thanks to Schrödinger for sponsoring this webinar and allowing us to bring the information to you. There were a few URLs and things that came out in the chat function during the course of the webinar. Please do grab those before the webinar ends, as always you won't be able to access them, or you may not be able to access them afterwards. And just a quick reminder that everybody will get a link to the recording of the webinar within a couple of days, and the certificate of attendance will come in that same email. If you liked this webinar, then do remember to check out chemistryworld.com/webinars to see what else we've got coming up. There's a whole variety of things, several with Schrödinger and a couple of other people as well. All that remains is for me to say thank you very much for joining us. I hope to see you again in another Chemistry World webinar. My name is Philip Broadus, and I'm the business editor of Chemistry World, and goodbye.